Okay, so now we're going to start out with a few lectures talking about cells. And a good way to start here is to talk about the principles of cell theory. And so there's three principles of cell theory that are described in your book, and I have a fourth one here for fun. Right, but let's talk about these ones in your book first. So the first one states that all organisms here on Earth, right, are composed of one or more cells, meaning that the body is at least one cell, right? Those would be our unicellular organisms like amoebas and bacteria, etc. And then, sometimes there are more than one cells, and so a good example of a multicellular organism would be a human, right? Composed of trillions of specialized cells, or an oak tree, something along those lines. The second one here states that bodies of the smallest organisms are single cells, and cells are the basic functional units of multicellular organisms. Sometimes you also see this written down as, something to the effect of the cell as the basic fundamental or functional unit of all life. And so that may seem like you're saying the same thing as the first principle, but it's not quite the same. It's kind of expanding upon it and talking about cells being the basic functional units of life. And then the third one, all cells come from pre-existing cells. And you can trace that all the way back to, the first living cell. And we're not covering that here in these principles of cell theory. We may never know exactly how that first living cell came into being through, some non-living process, right? Some non-living biochemical process. But after that cell, right, all cells come from pre-existing cells, including all the ones that we know of today. Now, sometimes you also see a fourth principle, and that's that each cell maintains homeostasis, and we already discussed what homeostasis is, right? Maintenance of relatively constant internal conditions within the body, right, despite what the external conditions are. Now, remember, we looked at this during the first lecture. These are your different levels of biological organization, going all the way from the level of the atom up to molecule, and then to cell, the smallest unit of life, and then proceeding all the way up from there to the level of the biosphere. So this is where we are right now. This is a nice picture from the book here, and it's showing you just sort of a comparison. These aren't actual photographs or anything, right? This is just an artist's interpretation of things, but based on scientific evidence and based on photographs. So here is just part of a generalized animal cell, right? So you can only see, what is this? Maybe like, imagine that you expand this all the way out off the screen, and maybe that's like, 20% of the cell, 30% of the cell, something along those lines. And then compare that to some of these bacteria, right? So some of you guys might have heard of Staphylococcus before, right? Little coccus bacteria shape, right? They're little spherical cells, and Staphylococcus tend to hang out in little clusters like this. E. coli, right? Escherichia coli. They're little rod-shaped bacteria, right? So here we are right here. This is a cyanobacterium, and I'm not sure if any of you guys know anything special about cyanobacteria. Some of you guys might, but these are photosynthetic bacteria, right? That are at the base of the food chain, just like, plants are. And these are really important. You can find some of these cyanobacteria out in wet, moist terrestrial environments, but most of these guys are going to be found in aquatic environments. And so this shows you that within the domain bacteria, that's where all three of these guys are, right? Representatives of our prokaryotic cells here. There's a really wide range in size variation. Staphylococcus are pretty small. Cyanobacteria are much larger in comparison. But these guys pale in comparison in size to, the average eukaryotic cell size. And then even though viruses are not officially considered to be alive, right, we still have them on here for a size comparison. You can see how tiny they are even compared to a small bacterium. An individual coccus bacterium. Now you can see something on here, right? So we have this little bar up here, and it says that the length of that bar represents 1 and this weird funky letter, and then M. Well, M stands for meter, right, which is a little over 3 feet. But what is this, the Greek letter right here? This is the letter mu, and that represents 1/1,000,000th. So this is 1/1,000,000th of 1 meter. So if you took that meter stick, right, imagine one of those just a little over 3 feet, and you divided it up into 1,000,000 equally sized units of length. That's what we're talking about right here. So that gives you an idea of just how small cells are. right? And then we have sort of a kind of a a typical size range down here. So eukaryotic cells, even though there are some that are smaller and some that are larger, right? Typically they range, from about 10 to 100 microns in size. By the way, this Greek letter mu, together with that m, this is the prefix micro. So this is a micrometer. Some people say a micrometer, or some people just shorten it to micron. So a micron, or a micrometer, right, or a micrometer, that is 1/1,000,000th of 1 meter, right? So most people just shorten it to microns, right? So this is 10 to 100 microns in size for prokaryotic cells. Even though there are some that are larger than this, right? Typical range in size from about 0.2 to 10 microns. So 0.2 would be what, 20% of 1/1,000,000th of 1 meter? Pretty tiny. And then look at viruses just for this fun comparison. 0.05 to 0.2 typical size microns in size, even though some of them it can be larger than that. And so, 0.05, what are we talking about there? That's 5% of 1/1,000,000th of 1 meter, right? So we're talking about something incredibly tiny. Now, the next few slides will just cover some basic cell terminology. First of all, we have cytoplasm. The prefix cyto means cell. So we got cytoplasm and cytosol here. What's cytoplasm? Cytoplasm is all the cell contents outside of the nucleus or nucleoid region. And so you might be thinking, well, wait a second. We already talked about a nucleus. What's a nucleoid region? Well, prokaryotic cells, recall, they don't have membrane-bound organelles, and the nucleus is a membrane-bound organelle. Okay, so they don't have that, but they do have a circular chromosome, and it's just within a part of the cell they call the nucleoid region. So having both of these in this definition makes it good for both prokaryotic and eukaryotic cells. So again, the cytoplasm, all the cell contents outside of the nucleus, if we're talking about a eukaryotic cell, or outside of the nucleoid region, if we're talking about a prokaryotic cell. Now, what is the cytosol? Cytosol is the fluid portion of the cytoplasm. So we look down here in this picture, and here's a phospholipid bilayer with a bunch of little molecules embedded in it, all these different kinds of proteins embedded in it. Some of these proteins have carbohydrate groups attached to them. We'll talk about that in a future lecture. And then we've also got cholesterol embedded here in the cell membrane. That means that we're looking at an animal cell here. And then here are the materials outside of that cell, and here's the stuff inside the cell, right? And you can see that term, cytoplasm. You might have noticed another term here, extracellular fluid. What does extracellular mean? Extracellular means outside of the cell. Here, this term, this prefix extra means outside of. So extracellular, outside of the cell. Kind of like extracurricular activities. If you ever participated in any extracurricular activities, right? They were, activities like band or sports or whatever that were outside of the regular curriculum, right? Doing these things after school, let's say. So here we go, extracellular, outside the cell. So what's the term that you often see for inside the cell? Intracellular. Intra means within or inside of, right? So here's everything in here within the cell is intracellular. Everything out here is extracellular material. Just a little recap, right? Remember that we have these two basic cell types, prokaryotic and eukaryotic cells. We talked about them in the first lecture. But yeah, so just a little recap again. Prokaryotic cells are much simpler than eukaryotic cells. And you can just see that here in this picture, right, compared to, this generalized animal cell down here. A lot more going on. This looks like a city, right, with taxi cabs and skyscrapers and people yelling and everything, and dogs running around everywhere, billboards. And this looks like kind of like a dried up old ghost town somewhere. Maybe there's an old guy sitting out on the porch or something. Anyway, right, so much simpler overall than eukaryotic cells. And remember, they have a single circular chromosome, so just one circular DNA molecule that is naked. It's not coiled or wrapped around any spools of protein, okay? And it's not inside of a nucleus. It's just in a nucleoid region. And, remember the nucleus is a membrane-bound organelle, right? So there's no nucleus and there's no other membrane-bound organelles. There's no mitochondria, there's no chloroplasts, there's no, Golgi apparatus, also called the Golgi body. There's no endoplasmic reticulum, no vesicles, no vacuoles, no anything like that. We'll talk about those things later on, by the way. Contrast that basic info there with the eukaryotic cell, much more complex. You can just visually see that. Much more complex than prokaryotic cells. And instead of one circular chromosome like a prokaryotic cell, what do we have instead? We have multiple linear chromosomes. Linear means like a line. It's shaped like a line. There's free ends. And why do I say multiple, by the way? I didn't mention this in the first lecture video, but multiple. Why not a strict number? Like in a bacterium or in an archaean, right, our prokaryotic cells, it's one. But why are we saying multiple? It's because, there is no strict number, right? The number depends on the species. Okay, so different species are going to have different numbers of chromosomes, or different eukaryotes are going to have different numbers of chromosomes in their nuclei. And here in this case, these linear chromosomes, they're all sort of kind of hanging around all over the place on top of each other here in this picture. We'll later on in the semester, we'll look at some pictures of them in a different form. Okay, but this DNA, right? This DNA is wrapped around proteins, right? And that's to help fit all this genetic material from these multiple linear chromosomes to fit in the space of the nucleus, right? So kind of like taking yarn or thread and wrapping it around spools. Okay, and so we'll take a look at that too at some point. And there are, yes, and where are those multiple linear chromosomes located? They are located within this membrane-bound organelle called the nucleus, right? So here's the nuclear envelope, this double membrane system that surrounds all that genetic material. And then in addition to that membrane-bound organelle, right, there's a whole host of other membrane-bound organelles. And I just put a few on here, right? Mitochondria, Golgi apparatus with a capital G, endoplasmic reticulum, rough and smooth varieties, right? And there's a lot of other things too. And we're going to talk about some of these big major kinds of membrane-bound organelles later. Now, this is a nice picture here from the book of a generalized animal cell. Now, generalized meaning not all animal cells are going to look like this. But if you just wanted one picture to kind of sum things up and give you an idea of kind of what an animal cell is all about in general, this is pretty good. So now we've got this one for an animal cell. We got this one for a plant cell. Both of these are eukaryotic cells. And then we've got our generalized prokaryotic cell of this bacillus or rod shape. And so, and you can see we got a lot of different things identified on here. Now, we're going to talk about most of these things, maybe not all of them, but anything that we talk about in lecture, be sure you can visually identify those things on one of these three diagrams. Okay, because on the test, I might put one of these diagrams, I won't tell you which one, but I might put one of these diagrams on the test and have you identify maybe, a few different things. Now you have a nice table here in the book. It's a pretty big table, and we're going to cover most of this information in here. So this is going to be a good summary table to take a look at every now and again. And the title of this table, what do we have here? Functions and distribution of different cell structures, right? And so see how this thing's organized here real quick. So we've got cell surface features that are important, how the genetic material is organized, and then we've got cytoplasmic structures, ribosomes, mitochondria, endoplasmic reticulum, central vacuole, right? All these different things. It gives a little breakdown of the function of each of these guys, and then it says whether or not they're present or absent in prokaryotic cells, and in our two most, our two groups of eukaryotes that are probably the most familiar to people, right? Animals and plants, even though we have other eukaryotes, obviously. We've already talked about some of those groups, right? But so we're not looking at those other groups here. We're not looking at the fungi, right? We're not looking at the different groups of protists, right? We're just saying prokaryotes, plants, and animals. Are these things present or absent? And a couple of these guys have a little, extra bit of information associated with them. So it looks like a lot of information, but we're going to pretty much cover this, and this will be a good summary. Okay. So let's start talking about some of these sort of cell surface features first. And the first one to discuss is the cell wall. What is a cell wall? It's a non-living protective and supportive layer secreted outside of the cell membrane of fungi, plants, some protists, and nearly all prokaryotes. Now notice what's not on there, right? So all fungal cells, all plant cells, almost all, right? Nearly all prokaryotic cells, and some protists, right? Like, different kinds of algae. But there, what does this mean, right? Who doesn't have a cell wall? Well, there are some protists that don't have a cell wall, like a paramecium or an amoeba, just for example, or our slime molds, right? They don't have cell walls. There are others too that don't have it. And then also, animals are not on here, right? So we are animals. That means our cells don't have cell walls. And let's just look at a few little examples here. So, nearly all prokaryotes have, right, a cell wall. And in bacteria, the cell wall contains a substance called peptidoglycan. And this peptidoglycan is composed of amino acids with carbohydrate groups attached, right? And so that's where you get this peptidoglycan. That's where you get that name from. The archaeal cell wall, in the group of organisms that superficially resemble bacteria but are genetically very different, right? They have a cell wall too, except it does not contain peptidoglycan. It contains other substances. So that's a physical difference between the two groups that's important. They have these underlying genetic differences, and even though they look kind of similar, there are some physical differences that have been found, and that's one of them. And if you're looking at a prokaryotic cell, right, where are we talking about this? Where is the cell wall? So here's the plasma membrane, right, that phospholipid bilayer that separates the intracellular components from the extracellular environment. And remember that the cell wall is this non-living protective and supportive layer secreted outside of the cell membrane, right? Outside the cell. So it's not going to be inside the cell. It's outside of that phospholipid bilayer. So it's colored here in this dark red color. It's only dark red so that way you can see it, right? That's how the artist drew it. Now this other stuff up on here, that capsule and slime layer and all that, we'll talk about that later. But right now we're just talking about cell wall. Hey, what about some of these other guys, right? Well, in plants, looking at this generalized plant cell here, right? There's the plant cell wall of and again, that's outside that plasma membrane, right? So it's on the outside of it, plant cell wall. In plants, the cell wall contains the polysaccharide cellulose, right? Which is a polymer of glucose. It's arranged in a particular way such that it's arranged into these tough fibrous strands. What about a fungal cell wall? Well, I don't have a fungal cell picture right here, but just imagine a eukaryotic cell that did not have chloroplast where photosynthesis take place, did not have plastids, or this central vacuole or anything like that, but had, quite a few of these other guys in here. And then it had a cell wall, but instead of containing cellulose, it contained chitin, right? Another polysaccharide. It's actually a polymer of modified glucose subunits. Glucose that has a little additional functional group added to it. Now I put this on here too. These guys do not have cell walls. These are a group of photosynthetic protists, right? That are called diatoms, and they live in freshwater and saltwater environments. These look like saltwater diatoms to me. They tend to have sort of more interesting and kind of complex shapes. But so they're unicellular and they're photosynthetic. And they secrete for themselves a shell that has a top and a bottom half that's made of silica, right? It's made of glass. And so again, this most people probably wouldn't consider this to be a cell wall. I've seen it, called just a shell before. It's got these two halves. And but still, this is also a supportive and protective layer that's secreted outside, and exists outside of the plasma membrane. So I thought I would just mention that. Let's talk about the cytoskeleton. Remember what that prefix cyto means, right? What does it mean? It means cell. And a skeleton, right? Well, you're probably familiar with the concept of a skeleton. We've got one, right? Composed of bones and everything. So does the cell have a bony skeleton? No, but it has an internal network of protein fibers that serve, several different functions. And so, and one of the functions is structural support, right? So, in that respect, it's kind of like a skeleton. So this is the skeleton of the cell, the cytoskeleton, right? So it's a network of protein fibers in the cytoplasm that's involved with regulating cell shape, holding and moving organelles, and that would be in eukaryotes because they have those organelles, involved in regulating cell movement and also involved in cell division. And it's found, the cytoskeleton is found in both prokaryotic and eukaryotic cells. And so here's a little artist picture right here where you can see this background network of all these different types of fibers. You can see we've got microfilaments, we've got microtubules, and we've got intermediate filaments. Here's some other things, right? This is a piece of the rough endoplasmic reticulum. There's a little vesicle. This is a part of a mitochondrion right here, where a bunch of ATP are produced. So that's all good and fine, but check this out. This is a photograph of a few neurons, and you can actually see that they've color-coded this on the computer because neurons aren't actually that color, but so that way you can see the genetic material here, the nucleus, but you can see all these red and blue fibers inside here. This almost kind of looks like cotton candy or something. So that's an actual photograph of the cytoskeleton. You can see how extensive that cytoskeleton is inside of these special cells. There's another view of that from a different book. Okay, so let's talk about prokaryotic cells. Prokaryotes were the first organisms to have arisen on Earth, right? And this occurred about 3.7 billion years ago. I just put BYA on here to shorten the amount of words on the slide, but if you have to write that out, be sure you know that it's billion years ago. 3.7 billion years ago, don't let anybody fool you. That's a long period of time. That's almost 4 billion years. Now, your book, this is a good point to make here, your book usually throws around the term, 3.5 billion years, and that's more so just for the sake of making it easy to remember, but something that's a little more realistic is about 3.7 billion years ago. That's when life is thought to have arisen first on Earth. And I've read some fairly recent papers that suggest that life might have even arisen as early as 4 to 4.1 billion years ago. So don't be surprised if you hear that one day. But for right now, let's go with this value, about 3.7 billion years. And remember that there's 3 domains of life, right? Bacteria, archaea, and eukarya. And two of those domains, right? Bacteria and archaea contain organisms that are composed of just one prokaryotic cell. For prokaryotic cell size, remember that most are very small, ranging from about 0.2 microns, right, to up to about 10 microns in size, even though some do get bigger than that. But so what does that mean? That means about 250,000 individuals, typical size, could fit on a period at the end of a sentence in one of your books. So that's how small we're talking about. Also, the typical human body contains about 100 trillion microbial cells, right? So here we were walking around thinking that we were just us. Prokaryotic cell shapes. Now, actually, there's a wide variety of shapes that are out there in the domains bacteria and archaea, but the 3 most common shapes are spherical, rod-shaped, and corkscrew-shaped like we see here. The fancier terms for these are coccus-shaped for spherical, bacillus for rod-shaped, and spirillum for corkscrew-shaped. Now, just for fun, these are the ones that I want you to know. And by the way, if we go back here to this diagram, right, that now you know what this bacillus means down here. This is a rod-shaped prokaryotic cell. So these are the ones I want you to know. But just for fun, right, this is from a different book, Foundations in Microbiology. And this just shows you a wider variety of shapes that are out there and how some of these guys, some of these cocci ones, how they how they cluster together in different forms and so on and so forth. And I thought I would just show you this. Oh, we have vibrios, curved rods, right? Some, ones that look like mold. We have spirochetes that are really curvy ones, a little more curvy than these spirilla ones. And anyway, so again, you don't have to know all the additional ones on here, but I just thought for fun I would show this to you. If you ever take microbiology, you'll probably dive into more of this. Let's talk about plasmids for a minute. So plasmids are small rings of DNA located in the cytosol of prokaryotic cells. They are extrachromosomal rings of DNA. What do you think extrachromosomal means? Right? Well, if extracellular means outside the cell, right, then extrachromosomal means outside of the chromosome, right? And so here we go. Here's that circular chromosome all wadded up where you can't really tell it's a circle. And then here's that plasmid right here. Extrachromosomal ring of DNA. So it's separate from that circular chromosome. And here is the interesting things about it, right? So there can be genes on these plasmids that are not located on the main chromosome, and these prokaryotes can get copies of plasmids from other prokaryotic cells. Could be other members of the population, right? Or sometimes even from other species, right? And these can contain copies of beneficial genes, like, for example, ones for resistance to particular antibiotics that maybe that individual didn't even have before it got a copy of this plasmid from somebody else. Right? Pretty cool. These guys have a few little tricks up their sleeves. So this is leading into our discussion of horizontal gene transfer, also called lateral gene transfer. So it turns out that prokaryotes, right, can exchange segments of DNA, right? And yeah, like I said before, this could be between individuals of the same species, also in some cases between individuals of different species, and it's accomplished through a few different mechanisms. But there's only one mechanism that I want you to worry about for this class, and that is conjugation. So in conjugation, right, two cells, like these two bacteria here, will connect so that genes, right, in the form of a copy of a plasmid can be passed from donor to recipient cell. So they're not both exchanging things like, oh, here, you give me some of yours and I'll give you some of mine. No, it's not like that. It's one cell is giving a copy of that plasmid to another. Now, how does this work on a basic level, right? Well, so what they call a conjugation pilus, some people call this a sex pilus, but a conjugation pilus reaches out and attaches to another cell. Then they're drawn close together, and then eventually a cytoplasmic bridge forms between the two, and one passes a copy of that plasmid over to the other. And after that, if that plasmid contains, a gene, one or more genes on there that individual didn't have before, now that individual has been transformed into a slightly different individual that now can maybe do something that couldn't do before, like be resistant to some particular antibiotic when before the antibiotic would have killed it. You don't need to know all the different steps here, but this is just a diagram of conjugation and kind of the rundown of it. You can see that circular chromosome here. Here's a copy of the plasmid, and so on and so forth. Here's the conjugation pilus, the cytoplasmic bridge, and so those things that I just mentioned, right, are shown here visually. Now, pili. What are pili? That's a plural term. A pilus is one, P-I-L-U-S. Right? Pili are protein projections from the cell surface of some prokaryotic cells. And, quite a few of them have it. And so we see, it's here in this generalized picture too. All these little things that look like little spikes. These are pili. And there's different types of pili, but the two that I wanted to point out to you are the ones that will end up covering, the individual, like you see here. These are attachment pili, also called fimbriae. These are small and found all over the surface of the cell. And the point of these things is that they help the cell attach and cling on to surfaces, making it harder to be washed away, let's say, or knocked off of a surface. Another kind is the conjugation pilus, right? So conjugation pili, like we just got through talking about, used in the process of conjugation for horizontal gene transfer. There are other types of pili too, but we're just going to leave it at this. What about locomotion in prokaryotes? Let's talk about that for just a minute. So here's some sort of general statements. A lot of bacteria and archaeans are going to just either cling on to surfaces or drift around passively in liquid, like out there in the water, not being able to move themselves, but that's okay because they're just drifting around and they're able to make a living that way. But about half of all prokaryotes are motile. That means if you're motile, you can move under your own power. A lot of these motile species are going to move around either using one flagellum or several flagella, right? Flagella is plural, flagellum is singular. Right, so here we just have a couple of different examples, right? This is a little rod-shaped bacterium here, where rod-shaped prokaryotic cell with one flagellum. Here we have tufts of flagella right here, one on either end. Just two different possibilities. And there are actually other possibilities too. Let's just go ahead and take a look at that. You don't have to know these names. So I just pulled this off of some website, right? But just to give you an idea. Some don't have any flagella, right? Some have one, some have a tuft on one end, some are covered with them all over their body, some have one on either end, some of them have a tuft on either end, right? So just different possibilities. Now, the structure of the flagellum, right? This flagellum is composed of protein fibers that are internal, and they make this thing whip around, right? And then it can generate current in the water and move the cell through the water column. Now, the structure of these prokaryotic flagella differs from that of the eukaryotic flagellum, and we'll take a look at that later, right? They're made of different proteins in a different arrangement. In addition to that, remember that even though bacteria and archaeans resemble each other superficially, genetically they're very different, and also, there are some structural differences. Like example, that structural difference that we discussed earlier in the cell wall in bacteria versus archaeans. Well, it turns out that in bacteria and archaeans that actually have flagella, the structure of the flagellum is different in both of those groups too. A big major difference is that the archaeal flagellum is thinner and made of different proteins than that bacterial flagellum. Okay, capsules and slime layers. I said that we would talk about this stuff a little bit later, and here we are. So that's visually represented here. Here in this picture, I like it. It kind of looks like a big, pink marshmallowy substance. Now, it's not, really actually pink, but that's just the way it's drawn here in this book. And notice where it is. It's outside of the cell wall. So what is this stuff, right? Well, these are protective coatings that are secreted outside of the prokaryotic cell wall, and they are composed of polysaccharides or protein. Now, what's the difference in a capsule or a slime layer? If you look here at this picture, you can think, oh, well, they must be the same thing. Maybe these are just alternative names for the same thing. And they're not exactly the same thing. The very similar idea in both, but a capsule is more well-defined, like you see here, whereas a slime layer is not well-defined. But long story short, right, they're there in the same spot outside the cell wall. They're there for protective purposes, right? They protect the cell from drying out, right? It just makes another gloppy, thick barrier of material that makes it harder for water molecules that are inside the cell to actually get through the cell membrane of the cell wall and make it through here to leave the cell, right? So it helps protect the cell from desiccation. That's a fancy word for drying out. And also helps the cell cling on to surfaces. So it adds another, amount of stickiness. What are biofilms? Fun little biology videos, right? Well, biofilms are also aggregations of one or more species of prokaryote to form a community surrounded by sticky protective slime. So it's a communal slime layer secreted and made by multiple individuals in a group of prokaryotes. And before I come back here and we go through the rest of this, what are we talking about? We're talking about this lovely stuff right here. Right, so here we have these, these might be spirillum. That kind of looks spiral shape, and these are pretty long to be bacillus. So I'd say these are probably spirillum prokaryotes. here in this bright green, and all this gloppy stuff is that communal slime layer that they all made. They're all living in it, right? Having a great old time. Right, so what does this do, right? Well, it helps the community stick on to surfaces and also protects it from drying out, kind of like the slime layer and the capsules do that we just got done looking at in that previous picture. It can help protect against antibiotics and disinfectants just by making more of a gloppy, thick, barrier for those things to be able to get through to actually make it all the way down to these cells. It also protects the cells from desiccation, like we had talked about before as well. And people that have studied biofilms, they found that there are actually little channels within the biofilm for the transport of nutrients down to other cells that might not be up near the surface, and also for waste removal of those cells that are down, that aren't near the surface, and so that way they can get rid of their waste. And so people who are studying this, I saw a talk one time, and this guy said, well, it's actually kind of like these cells are almost working together in a form of metabolic cooperation in a way, so that's interesting to think about. Have you ever seen a biofilm? Well, yeah, so dental plaque, right, that builds up in your teeth, that's a type of biofilm, right? So that's fun. And when you wake up in the morning, right, and you feel all that stuff all over your teeth, right, there you go, right? That's a biofilm. Also, biofilms can form, it talks about this in the book, can form on wounds, on surgical cuts, and on devices that have been implanted in the body that aren't naturally supposed to be there, like catheters, artificial hips and knees, pacemakers, right? And so think about a nice layer of biofilm, coating these things that you've put in your body.